Hi, I'm Tom. I'm going to talk about the mitochondrial assay we've developed at IBIS. So Steve introduced our technology. Les has gone through an introduction to how mitochondrial analysis is normally done. I'm going to talk about briefly the assay we've developed here at IBIS to analyze mitochondrial DNA. First I'll go through the format of the assay, how it's laid out, describe a little bit how we do the analysis. It's base composition analysis, as Steve has pointed out earlier. A little bit about how we process the data and do the analysis itself. Some work we did with blinded samples to do sort of a pre-validation at FBI DNA Unit 2. They're working on validating the assay for forensics use. The information content, how it compares to sequencing. Since we do base composition analysis, we get A, G, C, and T counts for PCR amplicons. We don't sequence them. How much information can we get from base compositions? How does that compare to if you sequence the whole region that you're looking at? What it looks like when we can detect heteroplasmies, mixtures. Les is going to talk in detail about analyzing mixtures. I'm just going to give a brief introduction to what the data look like. The sensitivity, it's similar to sequencing, and a little bit about the reproducibility we've seen in this platform with this assay. This is how our assay is laid out. It spans most of the control region around the origin of replication of the mitochondrial genome. It's a little over 1,000 bases that are targeted. We use 24 primer pairs that tile across. Here's HV1, hypervariable region 1, and we have 12 overlapping primer pairs that span that region. So you get a little bit of redundancy in information, which can be useful, especially in looking at mixtures, looking at heteroplasmy. You often get a corroboration between primer pairs. And then we have another 12 primers that span HV2. And these, as Steve showed with bacterial alignments, we can target highly conserved regions that span across variable regions. Most of the entire region of hypervariable region 1 and 2 is considered, well, it's called hypervariable for a reason. The variability isn't just scattered randomly, almost scattered randomly, but there are regions that are highly conserved. We can target those regions with the 3' ends of our primers and prime virtually everybody with all 24 primers. There's an occasional dropout that's very rare. And then we use the information in between to separate, to differentiate samples. This is the layout of one assay. There's 8 reactions. Each of those has 3 primer pairs in it. So these are shown grouped by color. What we do then is we group them such that each primer pair within a reaction that has 3 primer pairs, each one is as far apart from the others as it can get. And then we use a short extension cycle in the PCR. So these will only be a few hundred bases apart from each other, but if we do the PCR such that we allow very little time for Taq polymerase to synthesize a PCR product, then the synthesis doesn't have time very often to get from here all the way over to here, so that this primer will then make a product over here. So after a few cycles, you just get this and this and this. 8 is a good number for us because we use a 96-well plate format. 8 is a column, so you can get 12 columns on a plate. The product masses then are put together such that the products separate in mass space and don't overlap each other. Our products, as Steve pointed out, we have a length limitation if we're going to get unambiguous base composition assignments. So we keep them short as possible with some constraints. So we have one that goes up to about 150 in rare cases, 140 normally. The most of them are less than 100 or less than 130, around the 100 range. Then we modify the primer concentrations relative to each other to get some level of balance between products within a reaction. So there are several steps involved. First is PCR. And then we have to take and clean all salts out of the reaction to the best of our ability. And then we analyze them in time of flight mass spectrometer. We get raw mass spectra that are then deconvolved into masses, numerical masses. Those numerical masses then can be converted into base compositions. Those base compositions are used to develop a profile. That profile can be compared, like Les had said, a question to a known sample, a known to a question, whichever way it's supposed to go, or to a database. We can develop a database with this. So here's one PCR reaction. It has 3 primer pairs in it. Those generate a bunch of product. That's sprayed into a mass spectrometer. Well, before it goes into a mass spectrometer, first what happens is it's desalted. So we use a magnetic bead cleanup that Steve had shown the instrument and has an automated desalting. What happens within one reaction there is the crude PCR product with DNA and salts goes and is bound, DNA is bound to beads. Those beads have iron. They're pulled out with a magnet. And then with several wash steps with clean buffer, the salts are pulled off iteratively. And then you put a different buffer in. The DNA falls off the beads are pulled out, and the clean DNA is removed. That goes into the mass spectrometer. This is an example of one spectrum that has 3 PCR primers in it. So there's a lot of peaks. It looks complicated at first, but it gets very simple. So there's 3 primer pairs in each reaction, so you get a whole bunch of peaks. There's multiple states of negative charge on each of the amplicons that come up for each strand, forward and reverse. There'll be a minus 28, minus 29, minus 30, and both directions. For the forward and the reverse, for every product, that's at least 3 double-stranded products, so at least 6 strands with a whole bunch of peaks per strand. That's at least 6 DNA strands per spectrum. We can denoise that. And then we have these internal mass standards. We know exactly where they should be in a spectrum. Software can go and find those then. And as Steve had shown, there's actually more than 1 peak in each of those, so you get a very precise measurement of where those are. We know exactly where they should be using a computer. And then a calibration along the x-axis is performed. Peaks that represent any given mass then can be correlated to each other in a deconvolution using the standards to get a precise calibration of the x-axis. We can turn that very simply then into this. And what this is that. It's the same spectrum. It's just gone through an algorithm that deconvolves it into a series of masses. So each of these masses is then coded to the strand it corresponds to. So we just have mass numbers now, and these are in Daltons. Or if you had a mole of them, that'd be grams. Because we know what primers we put in, we can associate 2 strands into a PCR product. This and this can't go together with the PCR primers we put in this reaction, if that makes sense. So these 2 go together, these 2 go together, and these 2 go together. Now we have 3 double-stranded products. Those double-stranded products then, knowing that this is a product and these are its 2 masses, very simple computer algorithm can be used to derive a base composition for that. So this one has 40 A's, 9 G's, 40 C's, and 19 T's. Okay, so we can do that with these as well. We know a little bit more than that because we know the PCR primers we put in, we know how we selected them against an alignment of mitochondrial DNAs that's relative to a reference, and that reference then has exact coordinates. It's the Cambridge reference sequence. So a particular position in the Cambridge reference sequence can always be used to reference the beginning and ending of a PCR amplicon. So we can put an amplicon then with a base composition and coordinates onto a map, and that map is the control region of the mitochondria, okay, mitochondrion. So we have 3 within this reaction. That's the beginning of a profile. Each sample is split up into 8 wells, and each of those has 3 PCR primers in it. So we fill up, reaction 1 is here, here, and here, reaction 2 is here, here, and here, and it fills up a map. That map corresponds then to a list of base compositions, and what those, the information that's shown here is a primer pair. That's just for bookkeeping. It doesn't really mean anything. It's a number and a code in a database. This means something. This is, it's a little off, coordinates. So this is 464 to 603 relative to the revised Cambridge reference sequence, and a base composition. So a whole pile of base compositions in order of coordinates is a profile. 2 profiles can be compared directly then. It's just, you just align the coordinates together and you can say they're either the same or they're not the same. We can do 12 assays on one plate, so we'll get a whole bunch of raw spectra. These come out at about a minute per. We'll get that down to about 30 seconds per well. That turns into the same number of deconvolved spectra that go to masses, that go to base compositions, and then we get multiple profiles on a plate. So we can run, if you're doing databasing, for instance, you can do a bunch of these at a time. It's a little bit faster, and the analysis is a little bit easier than sequencing. So we'll get a bunch of profiles. To test and verify that we get what you'd expect when we do this assay, we have to verify that if you sequenced the mitochondrial region from a person, or if you did this base composition assay, would you get a predictable result that's consistent with what you'd expect from the sequencing? We, in collaboration with Armed Forces DNA Lab, AFDL, and then also the FBI, Les, and then Bruce. Actually, Bruce started this whole process about 6 years ago with a different, we were doing a different approach, but he came to IBIS and said, this is kind of neat. You could run some samples, see how you could do with. Later on, through the process, we developed an assay that can be used by people other than us. So we got 25 blood spots from AFDL, and we got 25 buccal swabs, cheek swabs from the FBI. Before getting the samples from the FBI, though, Connie had carefully looked at our primers and looked at alignments of known sequences for samples that had been sequenced, and carefully chose samples that should fool the system by making primer pairs drop up because they had mismatches in at least one primer throughout the system. And then with from NIST, from John Butler, we got 95 population reference samples that we tested the system against. All reactions are run on prefabricated kit plates. We kit them, and then all you do is add sample. You add 5 microliters sample per well, put them in a thermocycler, then you put them on an instrument when you're done. All samples that we tested for AFDL, FBI, and NIST gave complete profiles. There were no dropouts in this set. 3 FBI samples, though, were not pure single contributor templates. And this is where we first realized that we could use this approach effectively to analyze mixtures. We didn't know what we were looking at first. One sample was a blank swab. We didn't know that. We tried re-extracting it a few times and we found out it indeed was blank. We were not able to get anything off of it. With 2 of these samples, what happened was there were multiple products in several of the primer pairs. So we couldn't tell exactly what we were looking at first, but we could tell that we had 22 pure, what looked like single contributor samples, and then we had base compositions and a list from each of the samples that looked like they were mixed. And for each of those, we noticed that the profile from somebody else in this set of the other 22 was fully couched within the profile that looked like it had more than one contributor. We noticed that for both of them. It was a different sample from the set. And we also noticed that if you subtracted that out, what was left didn't appear to be in that set. And it turned out that was the case. What happened was a cheek was swabbed, and then the swab was given to somebody else that was married to that person. They were swabbed, and then the swabs were extracted and tested. So we were able to not only deduce that there was a mixture, but also what a potential contributor to that mixture was. And we saw several heteroplasmies, both C length and non-C length, and I'll show you what one of those looks like in a minute. When you sequence mitochondrial, or when you sequence anything, you get the order of the bases. Right? What we do is we don't get the order of the bases, we get the count of A, G, and C, and T. And we can take that count and we can compare a count knowing the beginning and end of a region between two individuals and say they're either the same or they're different in their A, G, C, and T count. If they're the same in their A, G, C, and T count, it doesn't prove that their sequences are the same. Looking at it the other way, if the sequence is different, you can't guarantee that the base composition will be different. It almost always is, but it doesn't have to be. Because one person could have a C to a T difference relative to person one, and then somewhere else within the amplified region, it could be a T to C, so it could go backwards. And then what you get compensating mutations, and you get the same base composition. So the question is, if we look at a bunch of sequences, what does the resolution look like relative to sequencing in terms of can we differentiate two samples or not? So using a little over 1,200 unique region sequences from GenBank for full mitochondrial genomes that have our complete region that we're tiling across in them, with each sequence differing at least by one base from everybody else, and ignoring C stretches because you can't always, with the search algorithm we use with base compositions, we assume the same thing as with sequencing. If there's length differences in the poly C stretches of HV1 and HV2, we can't guarantee that you won't see from two different tissue types, for instance, from the same individual, you might see a difference there. So we ignore that. We convert those sequences to base compositions and then cross-compare them. And we see that for 94% of sequences that could be differentiated, we can still differentiate them by base compositions. Now, because we actually amplify a slightly larger region than the minimum, now this isn't what you always sequence, but the minimum to be called HV1 and HV2 according to the guidelines for adding sequences to the population database that's used, well, at least the FBI population database, and a general agreement between people. The coordinates 16024 to 16365 in HV1 and 73 to 340 in HV2 are required for a forensics analysis. We amplify a little more than that, and using just base compositions, we can separate 94% of the sequences that we were looking at, whereas if you sequenced and got the order of the bases in these coordinates, you do 90%. So tiling across the full control region is a little more resolving than sequencing the minimum region. Of course, sequencing the whole region is a little more resolving than tiling the whole region. Now, Les had talked about what happens when you have two sequences within an individual, heteroplasmy basically. That's at least two sequences within an individual, and they differ by length. What you'll get then, at the point you put a primer down here to sequence, and tech comes in and you see the sequence as it synthesizes. At the point that it hits two different templates with different lengths, you get overlapping reads. If it's bad, it kind of flatlines and you can't read it. This is an example of a sample we had looked at. When we spray that in the mass spectrometer, using a primer pair that goes somewhere around here to here, it spans this region, what we see is multiple peaks that we can resolve the masses clearly from. If we correlate those peaks to each other, you'll see we can derive a base composition for these two here. Now, the mass difference between this peak and this peak is exactly the mass of a C, internal C within a piece of DNA. On the other side, the mass between these two peaks is exactly the same as a G. The G we use is carbon-13 enriched. Same here and here. Now, what we see there is when we get a base composition from these two, we see this composition, 44, 13, 42, 23. The next two peaks are 1 C longer, and then another C longer, and another C. So what you can't see clearly here, first of all, you can see that there must be a length mixture, but you can't read it after that. And coming back the other way, you can't read it. What we can see here is not only can we tell there's more than one, we can tell there's at least 4, and we can tell exactly what they are. And we can tell the relative abundances. So we can quantify them relative to each other. You'll notice the distribution of these, although the balance isn't perfect between strands, that's just PCR, it's not, nothing's exact. But you see the same distribution pattern, you can quantify that. Moreover, as you go and amplify here to here, the presence of this length difference doesn't deteriorate your signal for the next primer pair. They're completely independent events. This happened to be a sample we had looked at a long time ago, and we didn't know what we were looking at first. And then we measured it again, and we didn't know that it was the same sample until we correlated the profiles and saw, hey, you gave us that in 2003. This is one of the blinded samples. So this is what it'll look like in a profile. You'll see there's 4 products for one primer pair, and they all overlap. They're all within one spectrum, and they all differ by a C residue. For each of the poly C regions in HV1 and HV2, we have 3 independent primer pairs that overlap it, so you get a triple corroboration of your heteroplasmy. So you'll see it there, there, and there. Same thing here with HV2. You get 3 reactions. I'm a little off on the overlay. But you'll see there's 2 length variants clearly visible for each overlapping region of HV2. There's 3 regions. This one has 2 products per. Now that was length heteroplasmy. What does SNP heteroplasmy look like in this? You know what it looks like in sequence. It's 2 peaks with one peak being right on top of the other peak. What we see in the mass spectrometer is 2 peaks that are offset a little bit from each other. This is from another blinded sample. These 2 products, which differ by a C to T transition, they are in almost 50-50 abundance. If they weren't, you can still quantify. You can quantify the relative abundance of the 2. In the sequence for this sample, what was listed in the truth data that we received after doing these is an N at position 16176. Now you'll see this primer pair here spans 16102 to 16224, and that position is within there. And these are 16130 to 16224, and that position is within this one as well. So you'll see the heteroplasmy in 2 regions then. So you can corroborate, and you'll get the same abundance, relative abundance in those 2. And this is what you'd predict from it. It could be either a C or a T being added to the known base composition if you predict it from the sequence. Just from, let's see, 95 plus 47, I guess, that number of sequences, plus another 21 that we took from ourselves in the lab. We saw several different SNP heteroplasmies, excuse me. A bunch of C to Ts. The most common ones are C to T and A to G. And then we can quantify the relative abundances. I'm not going to go in gory detail to this table because you probably can't see it. But we see a series of samples that all have clearly resolvable SNP heteroplasmies. We see many more that have length. Length heteroplasmy is common in HV2. It's fairly common in HV1. And there's a region downstream of HV2 that sometimes is called HV3 that has both a dinucleotide repeat, a CA, and also a poly C region, and we see heteroplasmy in both of those. Sometimes there's a CA length heteroplasmy, and sometimes there's a C length heteroplasmy. As far as sensitivity at this point, many dilution to extinction experiments have been done with this assay at this point, because it's a common, it's a standard plate that Kristen makes to analyze system performance, the dilution series on it. But in coming up with how sensitive we could expect this to be routinely, dilution series were done, and we found that in general, all primer pairs within the triplexes had correctly and automatically detected the product that we knew should be there between 0.1 picograms and 6 picograms. And then if you do this many more times, you'll see that on average, you can expect full profiles anywhere from 1 to 10 picograms most of the time. Around 4 is pretty common, around 10 is pretty common. So we set a system performance criterion at a 25 picogram to be conservative and safe for a standard dilution plate that's used to assess and verify that a system that's about to be used to do an analysis is sensitive enough, the process is working, and the software is working. We've run a pretty high number of samples so far now, and using 3,300, a little more than 3,300 trials of the exact same template over and over at the same amount per reaction. This is 500 picograms per reaction in this analysis. That's about 2 nanograms total. You can use less, you can use more. If we've gone anywhere from unquantifiable to a known 37 nanograms per reaction with very little difference in output, at least visually. Using automated data analysis, that's 79,900 expected double-stranded assignments for this particular template does not have heteroplasmy. We got 3,298 full correct profiles, that's 99%. 33% of them had a primer pair missing for some reason or another. Sometimes it was not enough DNA got into a well. I mean, this is several thousand trials. Sometimes there was a minor miscalibration in a well that doesn't cause you to get the wrong answer, it just causes you to not get an answer. It's very rare. That was 85 total missed assignments, or 0.11%, and 113 artifact assignments, or 0.14%, all but 10 of which were easily correctable with the manual QC we normally do. Those are usually a salt addict, which we have to get salts efficiently cleaned out before they go into the mass spectrometer. And on occasion, you'll see what you can see is an extra peak after your product peak. And 21 misassignments, all but 12 were correctable with manual QC. That was 0.015% then. And the mass errors, what we call mass error, it's a measurement deviation from expected. It's measured in parts per million, as Steve had talked about earlier. It was around 10, so that's about what we can expect on average for this assay, about 10 parts per million. That's, what, a third of a Dalton for something that's about 120 bases long. So with that, I'll take questions if you have more questions. Okay.